Welcome everyone. My name is Andreas Lahner and I work for the Medical Genetics Center in Munich, Germany. And I will present you today in this lecture the variant classification system by the ACMG and AMP recommendations. But first of all, I want to thank the organizers and especially Dr. VORD for inviting me to give. So, what I want to do today is give you an overview about variant interpretation and the HUGO Education Committee, for which behalf I speak here today. And I want to introduce you to HUGO first. So HUGO, the HUGO Education Committee was founded only 1.5 years ago. And this happened because these three organizations, Human Genome Organization, HUGO, the Human Variant Project, HVP, and the Human Genome Variation Society, HGVS, merged only to one entity, and this is now HUGO. And as you can see here, this is a screenshot from one of the charters. The aim of HUGO is to develop, design, and guide the diverse global community for human genomics education and training. And as I said, you can find the homepage here. And especially I speak today as the head of the Variant Interpretation and Genome Database Subcommittee of the HUGO Education Committee. And I will not go to all of this text here, but our aim is actually, especially on my variant interpretation and genome database focus, to train molecular biologists, bioinformaticians, physicians, and lab staff. So this is our main focus, because we and HUGO think that the gap between developing countries and the first world, this is always sometimes referred to, the gap is even widening, and we do now in our lab, for example, whole genome sequencing, and we use advanced AE machine learning tools for variant interpretation. But we think that it's very basic and very important to train staff all over the world with state-of-the-art, free available tools, open source tools. And we are convinced that state-of-the-art molecular diagnostic is possible with a good trained staff. And this is what we have in focus. And as I already said, we have a broad representation of members all over the world. But I'm especially happy in your setting that I can present you Dr. Huido as also a member of the Education Committee. And he presents the Vitamins Organization for Rare Disease. And I'm happy that he is in our committee. Okay. This is the overview. Again, I start with HUGO, I already did, and give you some background on variant classification. Then I will go to the ACMG AMP classification system, which I cannot discuss in full detail, of course, because this can be this topic of a whole series of workshops. But I will show you how the system works, and I will give you some recent recommendations from the Sequence Variant Interpretation Working Group of how to use these rules. Okay, let's jump into it. So first of all, the ACMG uses a 5-tier classification system, which was actually published quite a long time ago from IARC, the International Agency of the Research of Cancer. And what they proposed at that time was to use actually a 5-class system. You see here the 5 classes, and each of the classes is linked to a verbal classifier, being pathogenic, definitely, likely pathogenic, and so on. But most importantly, these classes are linked to definitive probabilities of being pathogenic, and you can see the scores here. So these classes are linked to validated qualitative measures of causality. And most importantly, these classes then can be used to translate the genetic data to clinical data. If you test relatives for the variant, if you recommend surveillance for the family, or if you test in the research setting family members. So that the real gain of the system is that all the 5 classes are linked to clinical recommendations, to translate genetic data to clinical recommendations. And the goal of IARC in this sense is to give actionable clinical recommendations to genetic data, and this is, as we all may know, an accurate and consistent variant classification is a prerequisite for genetic diagnostic and also precision medicine in the future. So why are we speaking about variant interpretation and discrepancies? So this is a screenshot from ClinVar, and it shows you submissions with more than 2 submitters, and you can see here that most of them are concordant actually. or they are not clinically relevant, but a small part of them, 3%, are clinically relevant because they go over the classes 4, 5 to 3, 1, 2. So this means that people working in labs and looking at the exactly same type of evidence can sometimes come to different conclusions. And therefore, a robust and consistent standardized variant classification process is very important. An example is, for example, you find this variant in the gene and you might suspect that this is a pathogenic variant, ACMG class 4, because you got this information, let's say, from databases, the literature, variant interpretation software tools like Russell and Franklin, from other genetic boards or meetings. So my first rule and the most important rule in variant interpretation is that you never uncritically adopt an opinion or a classification from any other source. You always try to find the underlying evidence and, if necessary, interpret the variant according to all available data. Of course, there are exceptions, as I said here, for the databases, and I will show you examples later. There are very well curated data from LOVD or from the VSEPs, from the variant curation expert panels at ClinVar. And of course, these variants you can get for granted. These are ultimately classified variants. But for all other things, literature is full of false positives. And I will show you some examples of this automatic variant interpretation software tools. They are good and I use them, but I don't use them uncritically. I always check the codes if this makes sense or not. Okay, this is very important. And of course, you should use the ACMG AMP system, which I will show you in a moment. So, what is the ACMG AMP system for? So actually, it is to determine if a variant in a gene with a definitive role in a Mendelian order is pathogenic. And I marked here the term definitive because it's very critical that you must be sure that this gene is really causative for a disease. The aim was to reduce the number of variants reported as causative or pathogenic of a disease without sufficient evidence. And as I said, the databases and the literature are full of false positives. And this is very critical. What they provide actually is an evaluation of different data types, and we have 28 defined criteria, each with an assigned code. And these codes go to two different directions. In the pathogenic one, being supporting pathogenic, moderate, strong, or very strong, or in the benign direction, being supporting or strong. Some general considerations or disclaimers. As I said, these guidelines are for variants in all Mendelian genes. Rather, they are discussed in a Sanger sequencing assays or single gene or gene panel, exome, genome, or transcriptome. That's all the same. Okay. The important is that they are associated with Mendelian diseases. You should be very careful with candidate genes. These genes are also sometimes referred to genes of uncertain significance or GOS. And as of August 22, only about 4,680 genes are definitely associated with an human disease. So all other 15,000 genes roughly are not validated. Okay. Then the terms mutation and polymorphism should not be used. Instead, the term variant should be used, and this variant is then pathogenic, class 5, likely pathogenic, class 4, uncertain significance, a class 3, likely benign, class 2, or benign, definitely benign is a class 1. And most importantly, all variants should be reported using the HGVS nomenclature. You can find here the link. And as I mentioned already, copy number variations, mitochondrial DNA variations, and somatic variants, they have their own ACMG classification system, but it's not this one I present today. Okay, they have different ones. And to sum up, the pathogenicity of a variant should be determined by the entire body of evidence in aggregate. So this can be databases, literature, in-house databases, and so on, including all cases studied, not only the case in front of you. And you should arrive at a single conclusion for a variant, and not if it is causative or explains the phenotype in one individual and not in the other. So this is also very important. This is what the table looks like, and don't be afraid, we will not go to all of these wordings here, but just to give you an overview, Here are the different categories where the evidence can be looked for, let's say allelic data, segregation data, functional data, and so on. And here we have from far left to far right the scale from strong benign to very strong pathogenic. And it also comes with a combination table where we can sum up the evidence codes. So let's make an example. I have a variant which is very rare enough, absolute population databases. And the variant has been described or detected in a patient in trans with a clear-cut pathogenic variant. And you find a functional study which says that this variant indeed has a deleterious effect. Then I would have 2 moderate and 1 strong. And this would sum here 1 strong and 1 to 2 moderate in a class 4 likely pathogenic. So this is how the system works in the real life. As you might suggest, in real life there are also some problems. And these are here, for example, from an early study, the percentage of discrepancies of different labs using the codes. And as you can see here, some of the errors, like say population data and functional data, have a very high score of discrepancies. Why is this? Well, maybe you can see here from the wording, it says here, well-established functional studies show no deleterious effect. So who is to say what is a well-established functional study? Or here it says the minor allele frequency is too high for disorder. So what is too high? Who says what is too high? Or, for example, the other databases, the reputable source, of course, this should not be used anymore, but this is, of course, very error-prone. Who is to define what a reputable source is? So therefore, taking all these discrepancies into account, the SVI, and the SVI stands for Sequence Variant Interpretation Working Group at ClinGen, they modify the codes together with experts. So these are the experts. Everything which is marked here in purple here, you can see that these codes are refined by variant curation expert panels or clinical domain working groups. So they should define exactly what is too high and what is well established. And the SVI has already completed. These are the ones in red here, the ones in red. They have been completed by the SVI, and in green is marked all the criteria which will in the future be modified by the SVI. For example, these codes here, okay? To give an overview how it works, so these are the ACMG codes. Here, these are the variant curation expert panels, and they fill in the data. They are really the experts of the disease and the gene, and they can say, I know what is too high, and I know what is a well-established study, and I can give recommendations. And on this side here, we have the Sequence Variant Interpretation Working Group, and they support the expert panels in refining the tools, and they also develop general recommendations of how to use the criteria, their special codes. So in all these together, refine the ACMG codes further. Okay. I spoke many times about the Sequence Variant Interpretation Working Group, and you can see here the link. You just locate to the clinicalgenome.org, and then you work here, go here to Working Groups, and select the Sequence Variant Interpretation Working Group. Then you will see here all the general recommendations from this working group so far, and I will discuss some of them. And as you can see here, the chairs of the Sequence Variant Interpretation Group is Leslie Biesecker and Stephen Harrison. And Stephen Harrison I want to mention because I have many of the examples I show you today are from his presentations and from his YouTube tutorials. Okay, let's start with the SVI recommendation for the criteria PM2 supporting, which is pathogenic, moderate, type 2. And you can see here that there is a recommendation published from the SVI, a recommendation for how to use this criteria. So the SVI says that, as you might know, PM2M is for moderate. And a moderate evidence strength, as you can see here, represents an odds of pathogenicity of 4.33. This means that 4 out of 5 variants meeting this code should be pathogenic. And the SVI recognized that this is way too high. It's way too high. So instead, from a moderate strength, it should be used as supporting strength. So it should be downgraded from moderate to supporting. Okay. So this means that 3 out of 2 variants meeting this criteria are pathogenic. It was overrated in the past, but now in the future, you should always downgrade PM2 to supporting always. Okay. A second recommendation from the SVI came about the wording. So the standard original text was absent in population databases. But absent is absent really, should it be for dominant disorders absent, or should it be within the pathogenic range? So they suggest that it should not be absent, because for adult onset disorders like in EXAC and GNOMED, all the individuals are as a general population, and they are not a healthy control. It is clear-cut that you have for adult onset disorders or reduced penetrance disorders, you will have definitely individuals in the GNOMED database. For example, breast cancer, they are after the reproductive age, so you would expect to have variants there. And so this was this formulation from the SVI, and then the clinical working groups, so the clinical domain working groups, or the recent variant curation expert panels, so for example these ones, they were asked to fill in these numbers. So they were asked, so what is PM2 valid? And for example, here the rasopathy working group said, really it should be absent, so there should not be any allele count in GNOMED. And for example, the CDH1 working group said, well, we would allow 0.001% in GNOMED, and this would translate in an allele count of 1 to 2 alleles in GNOMED. And as you can see here, if you can go down to the recessive disorders like hearing loss or FIKU, you see that these numbers are even higher, so 0.007% or 0.02%, and higher allele counts are still allowed in GNOMED, and this would mean that PM2 supporting would be valid if it's in this range. Okay, so it's not absent; it's within the pathogenic range. Let's now take a look to case level data, and these are PS4, PS2, PM6, PM3, and PP4. So PS4 says the prevalence in affected is statistically increased over controls, and I will demonstrate it in a few seconds. So this should be used only for proband counting in a dominant setting. Okay, the example will follow in the next minute. And the PS2 is the de novo criteria. So it says de novo without confirmation or de novo confirmed. In that sense, it's moderate, and it's strong evidence. PM2 is for recessive disorders detected in trans with a pathogenic variant. So this should be used as a proband counting in a recessive setting. And PP2 says the patient's phenotype or family history is highly specific for a gene. So PP4, for example, should not be used for proband counting, but only if the gene is highly specific for the disease. And I will now show you some of the examples. Let's start with PS4. So as I said, PS4 says the prevalence is statistically increased, but as you may see from the wording, it's very hard in the rare disease setting to make such large case and control studies to come to a statistically valid number. So therefore, the SVI says that we would also allow it to count probands with consistent phenotype. So for example, if you identified in many probands with a consistent phenotype, you could use it with strong. If you identified in some patients, you can reduce it to moderate. And if you identified only in few, then it's supporting. And the exact numbers here, they should be given by the expert panels for the disease. I will show you an example in the next slide. So PS4 could be used for just a typical case and control study. You can sometimes find in the literature, or you can use it as proband counting for multiple unrelated probands with a consistent phenotype. And these are the numbers the variant curation expert panels come up with. So again, this is the rasopathy, PTEN, CDH1, and so on. And these are the numbers they come in for this difference evidence strength, supporting, moderate, and strong. And you can see here that, for example, for very rare diseases, you need 5 probants for strong, but for recessive cardiomyopathy, you need 15 unrelated probants for being a strong evidence code. And most importantly, the variant must, of course, be rare enough. So PM2 must also apply to use PS4. So if the variant is common, then you cannot use PS4. Now let's have a look at the de novo criteria, PM6 or PS2, whether it's confirmed or it's assumed. So assumed it's moderate and confirmed is strong. And here you must have must be very careful to apply this criterion only if the phenotypic consistency is high. So for example, and show you here the example with Cornelia de Lange syndrome in the nipple gene. So here the phenotype is highly specific for a gene. And if you find it in one case confirmed de novo, you can apply 1 point. If it's only assumed de novo, it's only 1 point, half. If the phenotype is consistent but not highly specific, you must reduce it to 1 point, 0.5 points. If the phenotype is consistent but not highly specific and a high genetic heterogeneity, let's say these are examples like seizures or mental retardation or non-syndromic mental retardation, then you must further reduce it to 0.5 and 0.25%. If the phenotype is not consistent at all, you should not use it at the de novo criterion, okay? And you can then take these points here and sum it up in this table below. And depending on the points you collected, you come from very strong to only supporting. And here we have the example for a de novo variant you find in the nipple gene in 3 bobans. Let's say this is the one you have in your lab. You made a trio exome, you may have confirmed de novo in 1 patient, then you have 2 points. And then you have in ClinVar and in the literature 2 citations. They say we found the variant, but it's not confirmed. So you have 2 points, 1 point here, 1 point here. You would sum up to 4 points, which would then be very strong, PS2, very strong. So this is how it would use. Now let's talk about prediction programs. They are also error prone and some people disagree of how to use it. These are the codes for PP3 pathogenic or PP4 for benign. And here I will show you the publication from Ghosh and Blon et al. A couple of years ago. Actually what they showed here with these 2 plots are in green are marked the meta predictors under different scenarios. I don't go into details, but just summary, what they find is that you should always use meta predictors. Meta predictors perform much better than single prediction algorithms. So this is the first message. And you see here that Revel, for example, is on different scenarios on top 1 position. We in our lab use Revel. And many of the other labs I know they also switched to Revel. So this was one recommendation a couple of years ago, that you use meta predictors, but earlier this year, this publication here came up from Sean Taftichian's and Mark Greenblatt's people, and they did a very nice work. They did a calibration of these different computational tools here. Maybe you know some of them, or maybe you use some of them, and I marked only Revel here. So they made a calibration and how to translate it in ACMG codes for the clinician recommendations. So this is a really excellent paper, and as you can see here, I marked Revel because we use Revel, for example, and you see here the broad range of the Revel scores over 0.932 to 0.003. And you can see here that this would translate to PP3 strong or even PP4 very strong, depending on the score. So normally in the normal setting, only supporting was able to be used, but now they suggest to broaden the use to strong, moderate, supporting, moderate, strong, very strong, depending on the scores you have here. And most importantly, they also give you a range. So for example, here between 0.290 and 0.644. So between these, you should not use it in any direction. So this is also very important. You see here in all tools, Everything which is in between here, you should not use it in any direction. Now let's talk about the SVI recommendations of using the criteria PP2. PP2 stands for supporting pathogenic 2 and says it's a missense in a gene with a low rate of benign missense variants, and pathogenic missense are common in this gene. And the criteria PM1, which is the mutational hotspot or well-established functional domain without benign variation. But let's first take a look at PP2. So the recommendations from the SVI was to use the gene missensed Z-score from the GNOMED, and I will show you how to retrieve it. This is calculated by comparing the expected versus the observed numbers of missense variants for a specific gene, and it's expressed as a Z-score. And the draft proposal from the SVI is if the Z-score is above 3.09%, then you could use PP2. Okay, so where do you get the message? This is a screenshot from the GNOMED browser, and I just put in GNOMED browser here, I looked for the gene, the KCNQ2 gene, and if you type in, you will see here this constraint matrix. This is here in large, and what you can see here is the missense constraint. These are the expected, these are the number of expected missense variants, and these are the number of observed, and this is then expressed in a Z-score, and you see here that the Z-score is 4.04, which is way above 3.09. So in this case, you could use PP2. Okay, this is the matrix how you find it. What about the hotspot criteria? So the wording is a mutational hotspot without a benign variation. And here we don't have an exact number, but I can give you the recommendations the SVI says. It says you should compare the expected versus the observed number for a specific region of a gene. So PP2 was dealing with the whole gene, and PM1 is dealing with a specific region of a gene. Okay? But again, you must be very careful because a missense constraint score could be highly simply because one region of the gene is highly conserved, but the rest of the gene not. And this would then be PM1. And how can we analyze this data? Again, I use the GNOMED browser for this, and in this case, this is an example for the SCN3A gene. You can look here, but then very importantly, you have to change the data set. Not GNOMED, but exec version 1.2. So you have to click on this and select exec version 1.2. Only in exec, you can see here this track regional missense constraint score. This is only available in exec. And what you can see here is that the gene is divided in 3 parts. This part here, which is not very constrained. This very small part here, which is highly missense constrained, you have an observed versus expected of 11%, and this part here, which is not constrained at all. So in this part of the gene, you have very few missense variant observed versus expected. And you can see here, for example, this is the ClinVar variance track for class 4 and 5, so only the pathogenic ones. And you can see here that class 4 and 5 variants cluster here in this region. So I would say that this region in the SCN3A gene is a PM1 mutational hotspot without benign variation. And again, if you want to have an explanation, here we have this question mark. If you click on the question mark, you have the explanation, and it says here that the missense constraint values closer to 0 indicate increased intolerance against missense variation. So this is closer to 0. But again, I cannot give you an exact number here. So the closer to 0, but I cannot give you a number. So at the end of the talk, I want to present you the recommendations regarding PVS1, which is for pathogenic very strong, and this deals with loss of function variants. And here we have this publication from 2018 or 19 from Tayoun et al. And the data I show you right now are from this presentation. And I strongly recommend it's free available, and I strongly recommend that you download it and put it on your desktop. So the first paper is that it is very important to determine the disease mechanism. So if you want to apply PVS1, you must be sure that loss of function is really the disease mechanism. And how can you do that? You can look in the ClinChen. I can show you right now how this is done. If the clinical validity is strong or definitive, or if not, then you can follow this other workflow, this other decision tree here. Okay? But where can you look for it? As I said, the determination of the disease mechanism is critical. So you can look at the ClinChen homepage at the dosage curation. Yeah? This is here the site. So for example, for the SEP2 gene, first of all, the gene is definitely associated with Mold-Wilson disease, and it is sufficient evidence for haploinsufficiency for Mold-Wilson syndrome. This means that if you have sufficient evidence for haploinsufficiency, then loss of function is indeed a pathomechanism. But you have also another possibility to look at it, and this is a screenshot from GNOMED. You can look at the loss of function metrics. So this is again the SEP gene in the GNOMED browser, and in this case I marked you here the loss of function metrics, and you see here the number of expected versus observed. You only have one observed but expected 44, and this translates to an observed versus expected ratio of only 11%. And if this threshold is below 35 versus 0.35, then it is a good hint that this gene is indeed loss of function intolerant. You can also use the PLI score. So PLI stands for probability of loss of function intolerance. And if this is 1, so 1 is the highest score, then also this is an indication for pathogenicity. So loss of function is PLI score is 1, the highest, and the OE score would be 0, the highest. Okay, and these two normally correlate. But you must be very careful again and keep in mind that the selection is largely blind to phenotypes emerging after reproductive age. So for example, you would expect to find pathogenic BRCA variants or APC or P10 variants, which are cancer susceptibility variants. They are late onset, and you will find them in the GNOMED database. Okay? And I will show you now an example of how we would do it for the splice variants. But again, I must stress how important it is that you answer the first questions, for example, for nonsense variants or for splice variants. And it's always the same, is this predicted NMD or not? For example, is the exon skipping predicted out of frame or is it in frame? And this is the most important answer for PVS1 to be answered for loss of function variants. I will give you an example how you can do it. Due to time limitations, I will not go into details. So for example, for this gene, the PKP2 gene, it is a splice variant, C.1379-1 G2A. And I want you to demonstrate how we would answer this question. So again, as I said, you should follow this decision tree. It's a canonical splice variant at a 1 or 2 position, and you should ask if it's out of frame or it's in frame, and if it preserves the reading frame. And how can you answer this question? So this is a screenshot from the UCC browser, but you can use it any browser. So this is exactly exon 6. You can see whole exon 6 here. And you can either count the bases, the nucleotides. So exon 6 is 132 base pairs long. And you can divide it to 3, and it's an even number. So it's in frame. So if exon 6 would be skipped, it would be in frame. But you can also look here at the nucleotides. It ends here with AT, but it starts only with a C. So as you might remember from the last exon and the next exon, this is in frame. Okay, so the exon skipping or use of cryptic splice site preserves the reading frame in this case. The second question is the role the region functional known or unknown? And I don't know about the role of the region here, but I can show you if you would locate to the gene in the GNOMED browser, and here you can see the gene is in reverse. So here's the arrow. So this is the last exon, this is the first exon, and we have 1, 2, 3, 4, 5. This is exon 6 in this end. And you see here, I typed, this is a search bar, and I typed in the variant 3.1379-1 G2A, here in this case, and you find it's in Clomid. It's marked here, and you find it 38 times. And you can see it, this is the variant here, and it's exactly on this splice site. But what's important here is that you see on these two isoforms, this isoform is missing this exon. And if you can see here on the expression, the most highly expressed isoform, this one, lacks exon 6. So this means that exon 6 is absent from the most expressed isoform, which is an indication that this exon is absent from biologically relevant transcripts. So in this case, the loss of function variants in this exon are frequent in the general population. On the exon is absent from biologically relevant transcripts, and therefore I would not use PVS1 at all. Okay, so this is an example of how you could look for PVS1 for this splice variant. And I mentioned this automatic variant classification tools, like for example VARSOM here, or this here would be Franklin. And I typed in here the variants, as you can see, for VARSOM and for Franklin, and they did not pick up this evidence. They say it's PVS1 very strong, and also Franklin says PVS1 very strong. So this is wrong. So this is not true. I don't say that VARSOM and Franklin are bad tools. I think they're excellent tools, and I use them very often. But as I said in my first rule in variant interpretation, is never uncritically adopt these codes, but you have to overrule it manually. Okay. So here, of course, VARSOM says it's BS2 strong because it's very common. I told you that it's 38 times in Clomid, and also Franklin says it's benign, maybe likely benign, but still they call PVS1 is very strong. But if this evidence would not be there, then you would maybe have a false positive here. So be careful with these classification tools. Always try to verify the evidence. As I said many times now, the SVI recommended not to use BP5 as a reputable source, BP5 and BP4 anymore. So don't use it. Reputable source does not exist anymore. Just throw it away. Okay, look for the evidence. And yeah, and now I finished with my presentation. To sum up, like every new method or technology, the ACMG classification guidelines need training and time. And I think if you train and get used to it, then you will have good results. And please check out the ClinChan resources for training material. You can go to ClinChan homepage, material and resources, and you find a lot of training material. And there's also a very beautiful YouTube channel from ClinChan where we have all the sources and many training materials available right now. And with this, I want to end and thank again the organizers for inviting me.